variations of the same molecular alphabet in an ant, in a whale, in a human. When you first hear this, you pause for a moment, because it sounds like poetry. But it's not a metaphor, it is the truth. Every living thing on Earth, without exception, carries a code written with the same four chemical letters: G, T, C, A. Guanine, thymine, cytosine, adenine. The four bases of DNA. These four bases combine in triplets to determine which protein will be produced. These triplets create a total of 64 different codons, and these 64 codes represent only 20 different amino acids. It's as if life learned to write a vast library of words using a very limited set of letters. This reveals the cramped yet creative nature of biology. This system is identical in every organism, from a bacterium to a human. Granted, there are exceptions. For instance, some mitochondria use different coding rules, but these don't change the fact that the system is largely fixed. It isn't surprising that we share a third of our DNA with a tomato. We are simply different versions of the same universal software. The great mystery: why these four? But here is the real question. Why were these four letters chosen? More importantly, why did it stay as these four? Theoretically, chemistry offered us many more options. Life could have evolved with other nucleotide sequences. Yet, evolution settled on these four, and since then, no living creature has violated these rules. None tried an alternative alphabet. Was this a necessity? Or was it the permanence of a single path chosen from billions of possibilities? Let's chase this question and see how these four letters occupy a strange place. Think about it this way. If life is the constant rewriting of an alphabet, then that alphabet doesn't have to be limited to four letters, right? And indeed, it doesn't. We now know, not just theoretically but experimentally, that life can be written with other letters. In the last 20 years, scientists have managed to produce alternative genetic systems that don't exist in nature, but closely resemble DNA. These structures are called XNA, xenonucleic acids. What exactly are they? They do the same job as DNA, they carry information, they replicate, and some even act as enzymes by catalyzing biochemical processes. But their internal structures differ. Instead of the standard AT and CG pairings, they use different base pairs and backbones. HNA uses hexatol instead of ribose sugar, making the molecule more flexible. TNA works with 3 O's, a simpler sugar, suggesting it could have been a more likely candidate in early Earth conditions. PNA completely discards the sugar phosphate backbone in favor of a peptide-based structure. This is a non-biological yet functional system. In 2019, a NASA-funded team created a DNA model with 4 new bases, Z, P, S, and B, in addition to AT and CG. They called it Hachimoji DNA, meaning 8 characters in Japanese. This synthetic DNA can form a double helix, remain stable, copy information, and is robust enough to potentially serve as the foundation for living systems. Life as we know it is not the only possible form. It's just our form. DNA is here not because it's special, but because it won the race. Where did these letters, adenine, thymine, cytosine, and guanine, come from in the first place? To find out, we must go back to the prebiotic world, where chemistry was experimenting with itself. Through the work of researchers like John Sutherland, we've seen that with just a few simple components, water, phosphate, hydrogen cyanide, and UV light, the precursors of DNA and RNA bases can be produced without any living system. Just heat, light, and the right sequence. In the early Earth, this energy was abundant. Ultraviolet light from the sun, violent lightning, and geothermal heat. The most fascinating part of these experiments is that instead of trying complex, separate paths for each base, a single potential pathway was designed where they could all form in the same environment. And it worked. These letters were achievable molecules. Whether via volcanic pools, hydrothermal vents, or organic molecules arriving on meteors, their emergence wasn't a miracle. It was chemically possible, environmentally plausible, and evolutionarily consistent. We must also remember that before DNA, there was RNA. For a while, RNA was the carrier of life. It could both store information and replicate itself, acting as both the content and the printing press. The fact that some viruses still survive relying solely on RNA shows this system once ruled the world. DNA came later because it was more stable. Mathematically, this process can be explained by statistics, molecular collisions, energy flows, and the random formation of chemical bonds. Given enough time and enough repetitions, the assembly of these four letters is not just possible, it's almost inevitable. The law of large numbers tells us that no matter how low the probability, if you repeat an event enough times, it will eventually happen. But there is a distinction here. Explaining something through probability doesn't invalidate the feeling it evokes in us. We humans don't live by logic alone. We live by meaning, intuition, and wonder. The fact that all of life relies on four tiny molecules joining in the right order is still a staggering reality. It's not just a math problem. Think of it like the first line of a poem. If those first molecules hadn't joined together, this sentence couldn't have been formed. This voice wouldn't have reached you. This thought would never have been born. Would you like me to adjust the reading pace of certain sections, or perhaps suggest some background visuals that would complement these scientific concepts?